Since December 2019, COVID-19, commonly known as coronavirus, has swept across the globe. With key symptoms like a dry cough, extreme difficulty breathing, and body aches, coronavirus poses a serious threat to our health.
Currently, coronavirus has caused 80% of infected individuals to have minimal symptoms, but 15 to 20% require hospitalization. With a 3.4% death rate, COVID-19 results in more deaths than the flu, which has an average death rate of 0.1% per annum. However, the COVID-19 death rate has proved to be highly stratified among different populations and countries.
Researchers at George Mason University have contributed to the mass frenzy to understand and eventually eradicate this pandemic. In their papers, Olds and Kabani analyzed nicotine as a potential enhancer of COVID-19 symptoms. While not as widely covered, nicotine usage claims 7 million deaths per year around the globe, with the CDC predicting more than 8 million people a year will die from diseases related to tobacco use by 2030. On average, smokers die 10 years earlier than non-smokers due to diseases like lung cancer, chronic obstructive pulmonary disease, and tuberculosis.
I'm Julia.
I'm Tyler.
I'm Ritika.
And in this video, we'll break down Olds and Kabani's findings to their essential question: is nicotine exposure linked to cardiopulmonary vulnerability to COVID-19 in the general population?
In their paper, Olds and Kabani analyzed the effects of coronavirus on individuals who were exposed to nicotine prior to their exposure to the virus. We predict, they report, that these individuals are primed to be at a higher risk because nicotine can directly impact the putative receptor for the virus ACE2 and lead to deleterious signaling in lung epithelial cells.
Let's break that down, first by analyzing why nicotine can increase the risks of COVID-19 susceptibility. Nicotine, or 3,1-methyl-2-pyrrolidinol-pyridine, is a toxic chemical found in cigarettes and Juul pods. Extracted from the plant Nicotiana tabacum, nicotine acts as a natural alkaloid. This gives it its bicyclic structure with a pyridine cycle and a pyrrolidine cycle. With the chiral carbon here, nicotine has two enantiomers. Enantiomers are a type of stereoisomers that are mirror images of each other but are non-superimposable. Just like your left and right hands, they are suited for different tasks and can potentially cause harm when used incorrectly. While nicotine only exists in the sinocer formation, the effects of it are even more so.
Nicotine is toxic because it acts as a teratogenic agent, or an agent that affects the development of a fetus, a neurotoxin, which causes cell damage and loss of synaptic function in the brain, and most importantly, an immunomodulator. An immunomodulator is the key reason why nicotine increases smokers' chances of having severe effects of not only COVID-19, but also of lung cancer and other diseases. It changes the level of antibodies and white blood cells in the body, which are important to keeping your immune system functioning and effective.
According to Ravathi Kumar et al., nicotine treatment dose-dependently limits the production of critical pro-inflammatory cytokines. Pro-inflammatory meaning causing inflammation, and cytokines meaning a type of T cell that exists in the immune system. The immune system is composed of different types of T and B cells, such as lymphocytes and macrophages. When the body encounters a foreign object, the immune system sends out different types of T and B cells to either inflame the area, attract other T and B cells, increase the temperature, and kill the foreign object, or go and destroy the object entirely.
When nicotine limits the production of these cytokines, the immune system is compromised and the effects of the foreign object or disease are compounded. Chronic exposure to cigarette smoke or nicotine eventually causes T-cell unresponsiveness. This apparent T-cell anergy may account for or contribute to the immunosuppressive and anti-inflammatory properties of cigarette smoke and nicotine. Nicotine-induced immunosuppression may result from its direct effects on lymphocytes, indirectly through its effects on the neuroendocrine system, or both.
Within the cells, nicotine has been shown to cause shortfalls in DNA concentration and content, compensatory elevations of total protein to DNA, and reductions in the membrane to total protein ratio. By impacting DNA, nicotine directly impacts what proteins are produced, how functional they are, and how well the cell can respond when the body is under fire.
Now that we understand nicotine and its harmful effects on a healthy body, let's analyze what cardiopulmonary vulnerability means, both in general and at the biochemical level. With cardio meaning heart and pulmonary meaning lung, cardiopulmonary vulnerability refers to any weakness associated with the heart-lung complex. The heart sends deoxygenated blood to the lungs, which then import oxygen into the blood through respiration. That oxygenated blood is then pumped to the rest of the body by the heart. If either of or both of these major organs have issues, the rest of the body is immediately impacted.
Illustrating lung diseases is the best way to explain cardiopulmonary vulnerability. Pulmonary arterial hypertension, or PAH, is a disease where obstruction in the small arteries of the lung causes an increase in blood pressure in the vessels. This increased pressure causes difficulty taking deep breaths, resulting in fatigue and lightheadedness. Idiopathic pulmonary fibrosis, or IPF, is characterized by lung scarring by an unknown cause. This scarring obstructs alveoli, making it difficult for sufficient levels of oxygen to flow from the lungs to the heart. And finally, chronic obstructive pulmonary disease, or COPD, is a condition of chronic obstructed airflow from the lungs. This results in inflammation of the lining of the bronchial tubes and excess mucus production. COPD is associated with destruction of bronchioles as a result of damaging exposure to cigarette smoke and other irritating gases and particulate matter.
Now that you've learned about COVID-19, nicotine, and cardiopulmonary vulnerability, the connection should be clear. Nicotine negatively impacts the heart-lung complex by reducing cytokine count, thereby increasing vulnerability for lung disease and its progression. It destroys bronchioles, thereby limiting oxygen absorption by lungs and transfer to the heart, and it changes DNA concentration and general healthy cell behavior, including mitosis, which can result in lung cancer.
With all of these traits, nicotine definitely increases the susceptibility of smokers getting COVID-19 and experiencing more advanced symptoms. This is Figure 1A from the Olds and Kabani paper. This figure details some of the biochemical occurrences that take place during a COVID-19 infection in healthy and damaged lungs. Now let's look at what nicotine does at a biochemical level.
Nicotine consumption results in a myriad of underlying conditions including hypertension, diabetes, cardiovascular disease, cancer, and immunological diseases. Accompanying these conditions are varied levels in physiological biomarkers resulting in anemia, leukopenia, raised globulin, thrombocytopenia, etc. These biochemical conditions, weakened immunity, and airway inflammation lead to an optimal environment for COVID-19 infection and proliferation. Let's see what physiological changes occur from COVID-19 alone in a non-smoking individual.
In non-smokers, coronavirus leads to an aggressive shortage of white blood cells with a shortage of lymphocytes. This condition is referred to as leukopenia with severe lymphopenia. In this condition, the individual is at a much higher risk of further infection. Alongside reduced white blood cell count, the quantity and functionality of T helper cells and cytotoxic T cells are affected by the virus. This is often attributed to alterations in the antigen-presenting cells that lead to the recognition of antigens by T cells and maturation of T cells to become virus-specific.
One of the most significant biochemical changes in COVID-19 patients, as is pictured in Figure 1A, is the downregulation of the sodium-potassium ATPase pump in alveolar epithelial cells. The sodium-potassium pump is an electrogenic transmembrane ATPase that moves 3 sodium ions out of the cell and 2 potassium ions inside of the cell against a concentration gradient. Within the lungs, the capacity to move ions across the alveolar epithelial membrane is essential for preventing fluid buildup. In the presence of a viral infection, ion transport channels such as the sodium-potassium pump and CFTR protein are downregulated by viral hemagglutinin, which clumps up red blood cells, and viral matrix proteins. This downregulation is caused by coronavirus alone in a non-smoking individual.
In smokers, nicotine is a detriment to the alveolar epithelial cells in the lungs. Type 1 alveolar epithelial cell, AT1, has the highest sensitivity to the oxidative nature of smoking. In patients that suffer from chronic obstructive pulmonary disease, there is an elevated level of apoptotic AT1 cells. When these cells are exposed to nicotine, their ability to fight against viral downregulation is compromised, thereby increasing the severity of coronavirus symptoms.
The immune system's ability to fight against COVID-19 is also dependent upon hydrogen peroxide and chloride anions. Hydrogen peroxide fights pathogens with inflammation. In reference to Figure 1a, hydrogen peroxide is a reactive oxygen species. This characteristic allows it to take on several roles in regulatory cellular events, including activation of transcription factors, cell proliferation, and apoptosis. However, high concentrations of hydrogen peroxide may result in negative effects to the cell, while catalytic suppression of hydrogen peroxide can lead to increased viral RNA expression and viral particle release. Thus, a proper balance is required for hydrogen peroxide to protect a healthy cell. The chloride anion works with the innate immune response mediated by phagocytes and neutrophils. Ramalingam et al. Showed that epithelial fibroblasts in hepatic cells expressed enhanced antiviral activity in the presence of increasing concentrations of sodium chloride. This suggests that resulting chloride anions regulate environmental toxins and viruses like COVID-19. Going back to Figure 1a, we can now understand that an exaggerated response by hydrogen peroxide and chloride anions can result in excess inflammation. Therefore, the molecules render the immune response ineffective and more susceptible to COVID-19 entry.
This resulting biochemical weakness can manifest as pulmonary edema. Pulmonary edema is an abnormal buildup of fluid in the lungs that can lead to shortness of breath, a common side effect of COVID-19. Reactive oxygen and nitrogen species can modify or damage ion channels, such as epithelial sodium channels, which alter fluid balance. Pulmonary edema is also a part of early pathogenesis of viral infections. One of the driving features in the pathogenesis of acute respiratory distress syndrome is the accumulation of fluid in the alveoli, which cause severe pulmonary edema and impaired oxygen intake.
With the excess responses of hydrogen peroxide and chloride ions, it's clear that their effects of pulmonary edema make a non-smoker susceptible to COVID-19. But let's take a look at a smoker's experience, specifically at the cellular and genetic implications of the nicotine receptor. The nicotine receptor is a ligand-gated ion channel that consists of 5 subunits and can exist in an interconvertible conformational states. When nicotine binds to the receptor, cations flow along a non-selective permeable opening in the channel and alter intracellular cascades. Certain subunit combinations allow for calcium ions to enter the cell. Calcium can lead to the activation of PKC in bronchial smooth muscle cells. This activation can lead to inflammation that causes bronchoconstriction, where bronchoconstriction is the tightening and thinning of bronchioles, resulting in difficulty breathing. Thus, the combination of calcium ions and hyperactive hydrogen peroxide and chloride anions greatly increases the effects of COVID-19.
With the CDC advocating that people wear masks, it's also important to understand how COVID-19 enters the cell. Research has shown that COVID-19 attaches to human cells through a spike protein at ACE2. ACE2 is a protein on the cell surface that serves as a door into the cell. The viral membrane fuses with the human cell membrane, allowing the genome of the virus to enter human cells. Researchers Zhang et al. Discovered that a post-2002 strain of coronavirus had mutated the spike protein to have a molecular ridge, thereby making it more compact. This causes COVID-19 to bind tighter to the receptor and infect human cells better and spread faster.
So how does this relate to the nicotinic receptor activity? When nicotine binds to the receptor, it alters the normal intracellular cascades. One possible alteration is the ACE2 expression in lung AT2 cells. Researchers Out et al. Established a definitive role of ACE2 in lung disease. They confirmed that ACE2 is a negative regulator of the renin-angiotensin system, which is a hormone system that regulates blood pressure, fluid, and electrolyte balance. In the setting of acute lung injury in response to the pulmonary infection with COVID-19, ACE2 can activate the right amount of inflammatory cytokines in apoptosis.
When nicotine binds, however, it changes the normal level of ACE2 expression, thus causing excess inflammation and allowing the virus to spread faster in a weakened host. Researchers Yu et al. Corroborate this. They found that cigarette smoke, or direct nicotine inhalation, inhibits the expression of angiotensin-converting enzymes in multiple organs and cell types. Therefore, smokers experience a compounded altered expression of ACE2 in compounded COVID-19 symptoms.
In conclusion, COVID-19 is a serious threat to the cardiopulmonary system and should not be taken lightly, especially by smokers.
Olds and Kabani show that COVID-19 and nicotine shut down ion channels responsible for regulating fluid, causing strained breathing.
They changed the normal levels of biomolecules like hydrogen peroxide, causing excess lung inflammation.
Nicotine alters ACE2 expression, allowing for COVID-19 to spread faster.
It's on this note that we stress the significance of social distancing and self-quarantining during these tumultuous times.
Stay inside.
Read a book.
And don't smoke cigarettes.